This is the second part of today's session in cognitive neuroscience on EEG. We will talk about how EEG is measured in the lab in this part of the session. If you haven't done so already, I would recommend you watching this video where you can see how an EEG recording is prepared and set up in an EEG cabin in an actual experimental lab at the University of Arizona. So here's a few things that are important about EEG recordings. So first of all, what we need is of course some kind of cap where we can position the electrodes according to a standardized system. So this system describes exactly where each electrode goes. Electrodes have names like FZ, CZ, PZ that refers to the position on the scalp, and by attaching them into the electrode holders, we can make sure that we're measuring from standardized positions. In the electrodes that you've seen in the video, just like in our lab here in Southampton, we have preamplifiers. So the signal is amplified right where it emerges from the head, and this has certain benefits. We will go into that in more detail later. So these so-called active electrodes have some features that improve the signal-to-noise ratio. Oftentimes EEG is measured in a shielded cabin to keep electromagnetic interference outside. And then one thing that is also very important in recording EEG is filling in this gel. So if you put on the cap, then there will be a gap between the scalp where the EEG signal emerges and the electrode. This gap is full of air and hair usually, and by using this gel we can bridge this gap to get a good electrical connectivity. Here you can see a participant in the lab which wears this head, and here you can see that the LEDs actually indicate how good the connection already is. So if it's green, the gel is in its right place. If there's one red one here, which means it doesn't have gel yet, and if there's some gel but the connection isn't right yet, there is kind of yellow, orange. So let's talk about the shielded cabin. So the EEG signal is very small deviations in voltage in the microvolt range. So a lot of electrical interference can occur due to that, because a lot of electrical signals are stronger than the EEG signal. So using an electrically shielded chamber, EEG chamber, is a good idea because it will increase our signal-to-noise ratio quite a lot. So what the shielded cabin really is a Faraday cage named after the British physicist Michael Faraday. So he discovered this principle that when you have some kind of electromagnetic field like here, and then you put a metal cage inside that cage, so this is some metal cage. A Faraday cage should have as few holes as possible, so the denser the metal is, the better. So of course you can see that the door is open right now, so in an open state this would not be a good Faraday cage, but if you close it, then here at the door frame connects with the actual door. There's some kind of metal mesh that will connect this, and then you have actually a perfect Faraday cage here. This is some kind of window, and there's also some kind of metal net in the window which closes the Faraday cage. So what happens if we have such a metal cage in a magnetic field? Well, there is ions in the Faraday cage, in any kind of metal, and the negative ions cannot, they will move according to the magnetic field. The positive ions cannot change. They're in a stable grid. but the negative, the electrons, they can change position, so they will go to the direction where the positive part is, which is here. And what this means is that there's an opposing magnetic field inside the cabin, which is exactly the opposite direction of the outside magnetic field. And in sum, this means the outside and inside magnetic field will cancel each other out, so there's no effect of the outside magnetic field in the interior of the Faraday cage. And this means this is electromagnetically neutral, perfect for measuring EEG. Okay, so we've seen that the Faraday cage allows insulation, which is good to reduce electrical noise. But another source of noise is actually still in the cabin, namely the participant themselves. So, for example, here you can see the EEG signal that we're measuring from one participant. And then here you can see this rhythmic, strong signal, which is much larger than the actual EEG signal, which would be maybe something here and here. So what this is actually a chewing participant. So maybe this participant has chewing gum. Another effect that the EEG signal, another thing the EEG signal is affected by is, for example, blinks. So, for example, here you can see this, that the participant blinks a couple of times, and this can cause a distortion in the magnetic field as well. So the eyeballs are little dipoles themselves. So the back of the eye, the retina, is more negative than the front, the iris. And in sum, what this means is that whenever you blink or when you move your eyes, there will be a deflection in the magnetic field, and the EEG signal, because it is suited to measure the very tiny deviations in the magnetic field that are the EEG signal itself, they will also pick up these blinks, and at these moments you cannot really tell the EEG signal from blinks apart. So ideally what you should do is tell participants not to blink during critical moments when the EEG signal is important to be measured, and also to, for example, not use chewing gum and to not move the head too much. This will induce noise on the electrodes. Another thing is sweat. So here you can see a drifting electrode, and this drifting electrode is changing voltage over some period of time. It can usually go for a few seconds up to a minute when this voltage changes, and this is because when you sweat, there's of course sweat is salty, and salt is a very good electrical conductor. So what this means, when you're slowly starting to sweat, then there might be changes in the current, and these changes occur over some period of time, not as fast as blinks, for example. It can be a few seconds at least. So you don't want that to happen either. So you could, for example, reduce the temperature in a shielded cabin so that it would not cause the participant to sweat too much. But of course it shouldn't be too cold, because you want the participants to feel comfortable too. Sometimes tricky to make participants feel well in the lab. Here you can see active electrodes. That's another good way of reducing noise and increasing the signal-to-noise ratio. So the principle behind active electrodes is that you amplify the signal as close as possible to where it emerges from the head. So imagine you have the same kind of signal going to an active electrode and a passive electrode. A passive electrode is really just a wire going from the scalp to the amplifier. Now what happens is, after that signal goes to the end of one of these electrodes, you have a larger signal here because it's amplified. It's not so large here. But could you just not amplify it here and then end up with the same thing? Well, the problem is that on the way from the tip of the electrode, where the signal emerges from the head, to the amplifier, there will be a lot of noise added. So this can be remaining electromagnetic interference. So Faraday cages aren't perfect, so there will still be something like electrical noise, for example, from electric engines, like an elevator, or maybe radio waves, this kind of thing. There's always some remaining signal that can affect the cable. But more importantly, the cable will also slightly move. So the participant will move a little bit, the cables can dangle down and so on. So this means that there's added noise, and what you will observe is then, if we're looking at the amplifier level, you have a large signal and some noise that is added here, but in total you have better signal to noise ratio than here, because no matter how you amplify the signal, you also amplify the noise that was added on the way to the amplifier. Okay, here you can see the raw EEG signal. So this is an EEG signal we're measuring from these electrodes over a period of time. 1 second is between two of these thin black lines here. And what you can see is what I've already shown you in the first EEG recording by Hans Berger, are these alpha spindles. So this 8 to 13 hertz waves, very rhythmic. So what probably happened here is that the participant closed their eyes during these periods. So when participants are in rested state and then they close their eyes, you will see an increased alpha, for example, here and here. And you can actually see that they blinked here. So maybe that caused this EEG to switch to more of an alpha level. And this is an eye movement. You can see this sudden onset and then sudden offset of the voltage change. This is because eyes move in saccades, so very rapidly, and then they rest for a while and they move rapidly again. Okay, so when we're looking at the EEG signal, it is interesting to see that there is actually different frequencies involved. So I've already told you about the alpha band frequency, and I've also told you that the frequencies that are involved in the EEG signal are a very important property of the measured signal. And so when we have an observed EEG signal like this, we can use a Fourier transform so that we can decompose the signal in each individual frequency. So in this example, you can see this is actually a composition of 3 frequencies: this low amplitude high frequency, this higher amplitude medium frequency, and this very high amplitude low frequency. So similarly, this is like how you can decompose chords into notes. If you're into music, you will understand that we're talking about frequencies here as well, of course. So a combination of frequencies will maybe look like this. Okay, the meaningfulness of these frequencies we will go into detail now. So the origin of different frequencies is that neurons are firing in temporal synchrony with each other, but each neuron ensemble has a typical frequency that is inducing these frequencies. So since, as I said earlier, we're measuring EEG the EEG we measure is the sum of very many different neural sources, then of course the composition of the actual EEG signal will have all kinds of frequencies. You don't need to know this in detail, but just to give you an idea that different frequencies are representing different cognitive functions. So, for example, the alpha band frequency, 8 to 13 Hertz, signifies a relaxed awareness. When you're tired, alpha is also higher. The suppression of alpha, so the sudden reduction of alpha, is in turn indicating that someone pays more attention. Gamma, however, high frequency, more than 32 Hertz, is related to multisensory processing and short-term memory. So there's different ways to measure the EEG signal. The spontaneous EEG is really just the EEG signal across a longer period of time. So you're measuring the voltage difference over maybe a couple of seconds, and then you can, for example, look at the frequency spectrum and say, okay, there's a lot of alpha going on, so the person must be sleepy. You can also use it for clinical research, so that certain patterns in the EEG signal indicate seizures, for example. The second way of looking at EEG data is event-related potential. So here we're not looking over a longer period of time. We're looking at a lot of short periods that are averaged together. And then this is always in relation to an event, hence event-related potential. So this is used to measure cognitive, motoric, or sensory processes. So here, for example, we have it for electrode P07. There's an event happening here. This is the ERP, like a typical shape you can see here. And this is the average across maybe 100 trials or so, whereas this is just like a single trial or like just a longer period of time. The different frequencies also characterize different phases of the sleep-wake cycle. So, for example, you can see that in when people are awake, beta and alpha bands are most active. When you're a little more tired, it's more alpha. If you're a bit more awake, it's more beta. And then in the first stage of sleep, you have low amplitude waveforms and theta bands. Then in stage 2, you have sleep spindles, the higher frequency and K complexes. So that's this one here. And then in stage 3 and 4, you have slow wave sleep, really high amplitude. If you compare this to, say, when you're awake, it's a much higher frequency, and this indicates a deeper level of unconsciousness. So these are delta bands. And then during REM sleep, which is associated with activity, and some individuals also with low amplitude sawtooth waves. They're not shown here, but this is related to a higher level of consciousness. So during REM sleep, that resembles wakeness a bit more, which is also interesting because during REM sleep, you dream a lot. Okay. So here you can see an example of how a seizure affects the EEG signal. So here you can see a relatively normal EEG, and then all of a sudden there's a very sudden onset here, and then you see this typical seizure and epileptic episode, and this can go on for 4 minutes, and with EEG you can exactly pinpoint the time when a seizure happened, and you can sometimes also find out in which brain areas, or at least which hemisphere, the seizure occurred. All right, so much about about measuring EEG. In the next part of this session, we will be talking about the calculation of event-related potentials, or ERPs.